All right. Okay. So as I was saying, I have posted up the stuff from Monday, and now the lecture I'm going over for FOXP2 is here, and the RNA folding homework is here. So if you open up this file, you will be able to print it out, and it has 4 problems on here. Part of them have, like, it tells you page 326 in your book, page 317, just to make it easier for you whenever you are going through it. The last page is really the only one you would have to print out to be able to cut and glue them together to make long strips. The images that it's telling you to look at in your book are the ones like this one here. So showing you, so it says problem 1, and it only has this one little bitty strip. And it tells you now, if an RNA molecule has the following sequence, that is the sequence that's on the strip of paper. If the 3D structure is treated with an enzyme that degrades single-stranded RNA, the following double-stranded sequence is obtained. And it has it listed out there. What is the 3D structure of the molecule? So if we go look at the sequence, we see AGCAU. So if we scroll through, AGCAU is here. So I suggest, just like I did with the other papers that you did, use a highlighter or something so that you can mark off where you're looking at. We go back up the other half of it. Now, paying attention 5' to 3', we need to read this one 5' to 3' also. AUGCU. You would use your highlighter to do this one. AUGCU, because this whole thing is 5' to 3'. So if you can't find it, make sure you're reading the right direction. So if you have this cut out and you have those 2 parts highlighted, you would be able to fold this piece of paper in half to have that A match up with that U match up with that A, that G would match with that C would match with that G, and that U would match with that A. And you could do just a little quick sketch of it on here to be like, whew, and show just some little binding in between. You have a longer sequence for this one, and I suggest using different colors. So when you find your UAA on it, because you have this whole thing out, looking for UAA, if you highlight this in yellow, then whenever you have your AUU and you have it highlighted in yellow, but you have the other ones done in like orange and green, and so if you can tell which one is going to match with which one, because you have to make sure that this one matches to this one matches to this one. So break them. So these are both yellow, these are both orange, these are both green. It'll make folding it in half a lot easier. Problems 3 and 4 have you make a big tRNA. Number 4 does not tell you, like how number 3 has these little boxes to tell you which ones are going to go together. Number 4 doesn't have that, but it follows the picture here exactly. So on the first one where you're told, like, okay, you're going to have 7 that match together, you can find those 7 and match them. You can find these. So number 3 will tell you these are going to be your 7, and you're going to find those and put them together, and you'll be making that structure on both of them. One of them tells you exactly which ones, the other you have to match them up based on what is shown properly. So there is the tip on here that in addition to the normal base pairing, G sometimes pairs with U. Inosine can pair with any of the 4 nucleotides. This tRNA has a variable loop of 5 nucleotides. Look on page 327 of the textbook for the clover leaf structure. That's this picture. So this variable loop here will have 5 nucleotides on it. So that'll help you out as you're working on this. So that is all of the information that you need to complete it. I do believe they show, like, they have like the size symbol, but I think on the book one, they just show it as P. for pseudouridine. So realize that if something doesn't match exactly like this, it's probably that whatever way they coded it on, I don't even know where I'm at, on here, like where they have an M, that just means there's a methyl group that's been added to it. And that's going to change the base pairing that it's allowed to do. You don't have to worry about it, just realize that's what that is. Again, remember that if you are not actively speaking, please mute your microphone. And if you ever have any questions, please feel free to unmute it to speak out. Sammy, it looks like you're not on mute. So we can hear somebody talking in your background. And if I mute you, you won't be able to take yourself off. That's the opposite of mute. Okay. So everything you need, you can see that that's not a very long worksheet, but I want you to practice them. All right. Sammy, I need you to mute yourself or I'm going to kick you out. All right. So there's not very many questions that you have to answer on here. The other thing I want to point out is because it's asking you what is the anticodon, what amino acid does it bring to the ribosome, that kind of thing. Because the images are shown on here as 5' wrapped around to 3', and 3' is where the amino acid is attached. Remember that when it binds, it's reversed for this because your mRNA is shown 5' to 3'. And remember, they're anti-parallel. If you have 5' to 3', the other has to be 3' to 5'. So looking now where this amino acid is attached, that's its 3' end, and it wraps, it's reversed through here, 3' to 5', and then goes up in the 5' on this side. So make sure that you are reading the anticodon in the correct direction to tell what the codon was going to be. And there's the codon chart is in your book and also in the PowerPoint here, 15.1. So that is everything that you're going to need to be able to do that worksheet to make sure that your answers are right. I'm going to pause.